Good morning, student. From today, we are going to start with the new chapter. That is chapter number 5, Origin and the Evolution of Life. Let us start with the chapter. Here, some part is deleted due to the pandemic COVID-19, and the remaining part, which are we going to learn, that is the Urey and the Miller experiment. Urey and the Miller experiment. Here, the Stanley Miller and his teacher Urey provide the first experimental evidence in support to chemical evolution theory of appearing. They designed a glass apparatus called spark discharge apparatus. Here, in this diagram, we see the one experimental setup which is said to be as the spark discharge apparatus. Okay, now the apparatus was first sterilized. The first thing is to be sterilized this apparatus and evacuated. Then the methane, ammonia and the hydrogen gas were pumped in the proportion of 1:2:2 into the glass chamber. Here, the very important is methane in one proportion, ammonia in 2, and the hydrogen gas is also in 2 proportion. And these gas are pumped into that particular glass chamber. A tube carrying water vapor. Also, the next setup is, the next provided is that they are provided with the water vapor. The tube carrying water vapor are also connected to that of chamber. Then the lightning effect was mimicked by electric discharge, carbon spark in the chamber. Also they give the electrical discharge. Now, process of evaporation and the precipitation. I repeat, the process of evaporation and the precipitation, that is PPT, was also stimulated by the use of heating mantle and condenser respectively. Now, what happened? The mixture of CH4, that is methane, NH3, ammonia and the hydrogen gas, that is H2, was exposed continuously to that particular electrical discharge for several days. Here, these are the glass chamber. These gases are introduced in this glass chamber. Carbon spark is also provided to that glass chamber. And here in several days, these electrical chamber is provided with the carbon or the electrical discharge, and they are exposed continuously to electric discharge for several days and causing the gas interact with each other. काय झालं त्याच्यामध्ये ते गॅस एकमेकांसोबत इंटरॅक्ट झाले means the chemical reaction takes place in that particular glass chamber, after which these were condensed. नंतर काय झालं ते गॅस कंडेन्स झाले. Then the liquid collected in U2 turn brown. जे काय लिक्विड त्या U2 मध्ये कलेक्ट झालं, ठीक आहे, वॉटर कंटेनिंग ऑर्गॅनिक कंपाउंड, दिस इज U2, U-shaped tube. Okay. The liquid collected in U2 turned brown. Chemical analysis of this liquid reported the presence of simple organic compound. And hence result guide, they show the presence of simple organic compound. For example, urea, amino acid, lactic acid, etc. This experiment strongly support that the just a minute. This experiment strongly supports that the simple molecule present in the Earth's early atmosphere combined to form the organic building block of the life. Okay. Combined to form the organic building block life. Okay. This experiment strongly support that the simple molecule present in the Earth's early atmosphere combined to form organic building of life. Okay. Here it is the experimental setup for the Urey and the Miller experiment. This is the glass chamber. Here in 1:2:2 proportion, they introduce the gases methane, ammonia, and the hydrogen gas. 1:2:2 proportion respectively. Then they also provided with the electrical discharge, that is the carbon spark, also added the water vapor, and several days this experiment was going on. And as the result or the end of the experiment, the gases get condensed, and in YouTube they form the brown color discharge or brown color, the liquid which is collected, which is brown in color. Okay. Now this is all about the Urey and the Miller experiment, and the result of this experiment is this experiment strongly support that the simple molecule present in the Earth's early atmosphere combined to form the organic building block of life. This is the main experimental or the result of that particular experiment. Okay. Now the second one is the RNA world hypothesis. Operant theory and the Miller-Urey experiment give us the understanding that pathway of origin of life on Earth goes from non-living to living. I repeat, they are from the non-living compound to the living one or the living organism. Like the gifts like necessity present of the necessity presence of stable genetic material and cellular machinery to carry out routine activity essential for survival. We are also aware that certain protein, which we call enzymes, catalyze the chemical reaction in the cell. We know that enzymes are the catalyst. They enhance or accelerate the reaction. We are also aware that the certain proteins, which we call the enzymes, which are the catalyzed, and the chemical reaction in the cell, they get accelerated to that particular reaction. It was in 1980 that these scientists independently found that the RNA can also act as a biocatalyst. These biocatalysts or these catalysts are RNA called as ribozyme. This catalyst RNAs are called as ribozyme, means ribosome and the enzyme ribozyme. For the discovery, they got in 1989 the Nobel Prize. For this discovery, this scientist is awarded with the Nobel Prize. This discovery provides important support for RNA world hypothesis. This hypothesis suggested that early life must have been based exclusively on nucleic acid, most probably RNA. What is the full form of RNA? Ribonucleic acid. And this discovery is also provided the important form of support for the RNA. That is, the hypothesis suggested that the early life must have been based exclusively on the nucleic acid, and the most probably that is RNA, because RNA is made up of ribonucleic acid. It was first proposed by the scientists in 1960, long before the discovery of the ribozyme. Fact in that RNA is found abundantly in living organisms, as we know. It is structurally related to DNA, and chains of RNA can evolve or undergo mutation, replicate and catalyze reaction. All support these hypotheses. In this RNA hypothesis, they found that RNA is found in all living organisms. It is structurally related to the DNA, and the chain of RNA can evolve or undergo mutation, replication, and catalyze reaction. All these reactions or all these terms are supported for that hypothesis. Besides, biomolecules like acetyl-CoA have a nucleotide in their molecular structure. Major evidences is extent of ribosome, the protein assembly unit. We know that ribosomes are the important cell organic which is responsible for the synthesis of the protein with the help of DNA, RNA. Okay, so they are called as a protein assembly unit in this cell. The ribosome translation process is catalyzed by the RNA. Here, the central dogma, that is the transcription, then the translation and the protein synthesis. DNA transcribe, RNA, that is the mRNA translate into the protein synthesis. This is the central dogma of that particular unit. Now, the next. These molecules might have undergone repeat replication and mutation forming varieties of RNA molecules with varying size and catalytic properties. Eventually, they might have developed their own protein code and machinery to survive the assembly of primitive cells. In due course, a double-stranded stable structure. We know that RNA is a single-stranded structure, but in case of rRNA, ribosomal RNA, sometimes they get folded upon themselves and form the double helical structure. But in case of DNA, we know that DNA is the double helical structure or double stranded structure which is very stable, and the DNA might have been formed and thus continued the ongoing journey which resulted in rich biodiversity on Earth. Okay. Now, this is the RNA hypothesis. RNA self-replicate. Here the RNA form the inorganic source, RNA catalyze protein synthesis, then here the membrane forming change internal chemistry allowing new functionality, and here they form the double stranded DNA structure, that is the DNA become master template. RNA codes both DNA and protein, and here the protein catalyzes cellular activities. This is all about the RNA world hypothesis. Okay. Now, in our today's lecture, we learn the two experiments. First one is the Urey and the Miller experiment, and the second one is the RNA hypothesis. Okay, Urey and Miller experiment, they, with the help of the apparatus or the experiment, they designed a glass apparatus and named them as a spark discharge apparatus. Here they use methane, ammonia and the hydrogen gas in 1:2:2 proportion. Okay, then the tube also carry water vapor and lightning effect. Then the mixture is continuously to electrical discharge for several times, causing the gas to interact with each other, and finally the liquid which is collected in U-tube turn brown. Chemical analysis of this liquid reported the presence of simple organic compound, that is the urea, amino acid and lactic acid. And this is the experimental setup of Urey and the Miller experiment. At the time और एक्झामच्या दृष्टिकोनातून ही डायग्राम इम्पॉर्टंट आहे. नेक्स्ट, RNA world hypothesis. In RNA world hypothesis, O'Brien and the Urey experiment give an understanding that the path of origin life on Earth goes on non-living to living. Okay. Also, they are aware with the certain protein which we call the enzyme, which is the catalyze the chemical reaction in cells. Then these catalyzed RNA they called as the ribozyme. For this discovery, the scientists got the Nobel Prize in Chemistry in 1989. This discovery provided the important support for RNA world hypothesis. The hypothesis suggests that early life must have been based exclusively on the nucleic acid, most probably RNA. Then, it was first proposed by the scientists in 1960, that long before discovery of the ribozyme. In fact, the RNA is found abundantly in all living organisms. It is structurally related to the DNA, and the chains of RNA can evolve to undergo mutation, replication and catalyze the reaction. Besides, biomolecules like acetyl-CoA have a nucleotide in their molecular structure. Major evidences in existence of ribosome, that is the protein assembly unit in cell, and in ribosome, translation process is catalyzed by RNA, and we know that nowadays we learn that is the mRNA, messenger RNA, is playing important role in the translation process. These molecules might have undergone repeated replication, mutation forming varieties of RNA molecules with varying size and catalytic property. Eventually, they might have developed their own protein code and machinery to survive an assembly of primitive or the starting or the first life on this earth. In due course, a double-stranded stable structure, that is the DNA helical structure, might have been formed and thus continue the ongoing journey, which resulted in rich biodiversity on Earth as we learn today. This is the diagrammatic representation of the RNA world hypothesis. RNA self-replication, RNA forms form inorganic. Sources, RNA catalyzed protein synthesis. Okay, now here the membrane formation, change internal chemistry, allowing new functionality, and therefore the protein catalyzes cellular activity. DNA become master template and RNA code both DNA and the protein. This is all about our today's lecture. In our today's lecture, we learn the Urey and the Miller experiment and the RNA world hypothesis. Okay, student, please do your own notes with the help of this PPT. स्वतःचे notes तयार करा, PPT पहा, diagrams ची practice करा. Most of syllabus जो आहे आपला 25% syllabus due to the pandemic delete झालेला आहे, त्याच्यामुळे तुम्हाला बाकीचा जो पार्ट आहे त्यासाठी व्यवस्थित अभ्यास करावा लागणार आहे. काही डाऊट असतील, काही प्रॉब्लेम्स असतील तर आपल्या व्हॉट्सअपच्या ग्रुपवर वगैरे तुम्ही शेअर करू शकता. Okay, this is all about our today's lecture. Stay home, stay happy. Thank you.